Hi everyone. In this video I want to talk a little bit more about gene regulation, talking about different levels of regulation and autoregulation and a couple other things. So let's start out with autoregulation. So far we've been talking about regulation where a gene gets either turned on or off. In particular, we focused on lactase and how sometimes lactase is all the way on and sometimes it's all the way off. But in other cases, we want to just kind of moderate the level. We want to make sure that we get the right level, not too much, not too little. So here's a picture of that on the top we want, or it would be optimal for the gene to produce a medium amount of protein, not a huge amount of protein. So how can we do that? Well, a very efficient way to do that can be to have a binding site on the same gene. So here we have a binding site for protein A, which means that this protein is going to come over and bind to this binding site and see it's a negative regulator. So let's think about how that's going to work. So if we have a low level of protein A, then we will not have ABS binding and we will continue to produce more A. However, if we have a high level of A, that should say we will have ABS binding. See here, a binding of A to the ABS, which will then lead to less or no A production. So if we think about what that means that we're going to get a medium amount, right? So we'll continue to produce protein A until we have enough that it comes over and shuts itself off, so we'll have kind of an intermediate amount of A. And so we call this negative autoregulation. So auto meaning self, so it's regulating itself, and it's negative meaning that it's negative. It's a negative regulatory relationship. And we can draw this like this, right? So that A is negatively regulating itself. So this is a common means of regulation. So here's an example that introduces a new concept. So far we've talked only about transcriptional regulation. Here we're going to talk about splicing regulation. So in the yeast S. cerevisiae and many other species, ribosomal protein coding genes regulate themselves at the level of splicing. Okay, so here we're talking about, here's the DNA, and we've drawn out a gene with two coding exons, ATG, another coding exon, and then an intron in between. And so our initial RNA looks like this, and then it can get spliced. So you see this intermediate region has been removed. It's been spliced to produce a protein coding region, or unspliced. So if it's unspliced, actually this does not encode a protein because it has a disruption in the middle of this intron. It has a stop codon in a way that actually causes this to be degraded. Okay, so we have this single intron that can either be spliced or unspliced, leading to either protein or to degradation of the transcript, no protein. And this decision as to whether to splice and make a productive RNA or to not splice is actually implemented by binding of this RNA protein. So now we have the encoded protein binding to the RNA and causing it to be more likely to be unspliced. So this protein blocks splicing of this transcript. So we can see that this is negative autoregulation, right? Because the more we have of this protein, the more it's going to cease production of more protein. It's going to block production of more protein. But the new wrinkle here is that this is happening. Notice this is not binding to the DNA. It's binding to the RNA and influencing processing, splicing of the RNA transcript. So we're starting to see now that there's kind of a whole zoo of different possibilities for regulation. So we've talked about transcriptional regulation, we've talked about negative autoregulation, and we've talked about regulation at the level of splicing. So there are actually a number of different possible outcomes of regulation. So one possibility is that we can produce two different mRNAs that have one is very long-lived and the other is short-lived. That means just that this mRNA will be around for a long time. It has a long half-life, whereas this mRNA gets degraded quickly. Well, what that means is that the long-lived one is going to produce more protein because it's around for more time, and the short-lived one will produce less protein. So this is another way of a subtle regulation of producing two mRNAs that both produce protein, but one of which is around for longer. And this kind of thing can happen by splicing differences or even promoter differences in UTRs, differences in polyadenylation, which we haven't discussed much, splicing of sequences with binding sites for degradation factors, all sorts of things. So my point here is not for you to memorize all this stuff, but to just get a sense that actually regulation is a very ubiquitous and diverse phenomenon that can happen a lot of different ways. So another thing is that we can produce two different proteins by a phenomenon called alternative splicing, which unfortunately we won't have time to talk about, I don't think, for now, and that these proteins can have different levels of activity. Like they both are enzymes that produce the same thing, or they're both transcription factors that bind the same sites, but one binds very strongly and one binds very weakly, or one enzyme is very powerful and the other one is somewhat weak. So this is just to say there are many different ways of regulating genes, and there are many different possible outcomes of regulating genes. So as we've also started to see, some regulation is very dramatic, it's on-off, and some is very subtle. It's just sort of tweaking slightly the level of protein production. Okay, so I think the last point I want to make in this video is that I'd like us to learn the different levels of regulation. So we've talked now about two different levels of regulation. So one is regulation at the level of DNA, at the regulation of transcription, right? So this arrow from DNA to RNA is transcription. And then we've also talked about at the level of processing the RNA, so splicing the RNA, the initial transcribed RNA to the final spliced and processed RNA. So we have this level of regulation, so we could regulate splicing, regulate processing. It's also possible to regulate the level at which an RNA is translated into protein, so that would be regulating at this level. And then finally, we can regulate at the level of, for a mature protein, regulating its fate. Okay, so we can call these different levels. This is the level of transcription, level of translation, and here we have splicing, as we said. And generally, we can call this RNA processing. And then finally, we have going from the initial protein to the mature protein, so protein processing, which you may have learned about in some of your other classes, where the initially produced protein is further modified by cleavage or addition of chemical moieties to lead to a mature protein. So we have all these different levels of gene expression. All these levels are necessary for gene expression, and all these levels can be regulated. And then finally, we have our last step, which is how long the protein is around. At what rate is the protein degraded, disassembled, and therefore ending the function of that protein. So we've talked about these two levels. We talked about transcription and we talked about splicing. So on this slide, what I'd like us to learn is that people talk about, a common way to talk about regulation is the level at which it's happening. So a transcription regulation we call transcriptional regulation. This splicing bit, or any part that's between the initial RNA production and the final RNA, we call post-transcriptional regulation. Regulation of translation is translational regulation. And then all this stuff here, regulating protein degradation or protein processing, we call post-translational regulation. Okay, so we have transcriptional, post-transcriptional, translational, and post-translational. So just don't get tripped up here. In fact, Of course, everything after transcription could be called post-transcriptional regulation because translation is post-transcription, right? But we are going to reserve this word. We're going to only use this word for this stage here between initial transcription and translation. Okay, so transcriptional, post-transcriptional, translational, and post-translational. So I'd like us to learn this stuff to build skills to identify what we mean by these four terms. So let's talk about a post-translational mechanism, which is called ubiquitin. So ubiquitin is this small protein. It's produced by a gene, the ubiquitin encoding gene. And this is a protein that actually is going to get attached by a chemical bond, by a covalent bond, to other proteins. So the way this works is like this. So this U will mean a ubiquitin, okay, a ubiquitin protein. So here we have a protein called A, and this protein will be fairly stable. It will continue to exist. However, if it gets bound to ubiquitin, if ubiquitin gets attached to this protein, that will lead to degradation of the protein. So you can see that addition of these ubiquitins is a way of regulating protein A, and it's at a post-translational level, right? This is a protein that has been translated, and now we're regulating whether or not it gets degraded. And we can in fact regulate this process. So some protein B will be involved in determining whether ubiquitin does or does not get attached to A. So B can regulate ubiquitination of A, and therefore it's regulating degradation of A because it's causing ubiquitin to be added or not. And so just like any other regulation process, just like transcription or splicing, here we have another factor that's acting to regulate the gene, one protein regulating expression of another gene and protein. And so we can work this through. This would mean if B is present, it causes U to be added, ubiquitin to be added, and protein A to be degraded. If protein B is not present, then ubiquitin does not get added, and we have a lot of protein A because protein A does not get degraded. Or we can have a negative regulation where protein B is blocking ubiquitination of A. So without protein A, ubiquitin gets added, and we have little protein. Whereas in the presence of B, it blocks ubiquitination, and A stays stable. Right? So just like we saw in transcription, where we can have a protein influencing transcription either positively or negatively, we can regulate ubiquitin either positively or negatively. So which level would we call this? Well, again, we're talking about regulation of a protein actually being degraded, so way down here. So this would be an example of post-translational regulation. So one more point to make is that that this level can be kind of multi-level regulation. So alternative splicing can lead to RNAs that have different levels of degradation, or RNAs that have different levels of translation, or to proteins with different level of degradation. So here we have differences in splicing, so that's post-transcriptional regulation, which is going to lead to different fates at the level of how long the mRNA is around to translate, how much it's translated, or the fate of the protein. And so things can get complicated is the main point here. And you'll see as you move on learning more genetics and more cell biology, you'll learn examples of this. And so it's good to be on the lookout if we can kind of have a scaffold of these sorts of things happening. Then we have a way of understanding new cases, new interesting cases that we learn. So key concepts from today are autoregulation, the levels of regulation, and ubiquitination. And then the other thing I would point out is regulation of splicing. Thanks very much.